He's again a member of faculty from my university, and I would also like him to restrict himself within 10 to 12 minutes. Dr. Syed Sikandar Azam is a member of Pakistan Academy of Sciences, and he has established a computational biology lab at National Center for Bioinformatics, Quaid-e-Azam University, Islamabad, where he's serving as an associate professor. He has produced 92 research publications. He is recently awarded by Ernest Match Fellowship Award from the Austrian Science Foundation. He has research experiences, particularly in computational chemistry and molecular dynamics, simulation of protein complexes to understand their structural and dynamical aspects and role in biological systems. Recently, one of his researches on COVID-19 was published in European Journal of Pharmaceutical Science and has discussed some of this finding in his presentation. So I would now request Dr. Sikandar Azam to present his today's talk. Over to Dr. Sikandar.
Hello, can you hear me?
Yes, we can hear you.
Thank you so much, Professor Dr. Mohammad Ali, the worthy Vice Chancellor of Quaid-e-Azam University, for a nice introduction. Today's topic of my talk will be the design of a novel multi-epitope based vaccine for pandemic coronavirus disease, COVID-19, by Vaccinomics and probable prevention strategy against Avenging Zoonotics. This is one of the, next slide, please. This is the first research article which published from Pakistan in European Journal of Pharmaceutical Sciences. Before that, going into that detail, I will discuss about the where I belong to, the computational biology lab of National Center for Bioinformatics. So computational biology is actually the field in which we use to extend our knowledge of science by using various computational techniques. And as far as the computer speed and capacity have paved a way and opened new horizons for different fields, same is happening with the multidisciplinary fields as well. So the outline of my talk will be the computational biology overview and our research highlights, vaccine and drug design with the recent trends, and molecular dynamic simulations of those targets, along with the prevention strategies and acknowledgements. What we do actually, computational biology is an amalgam of various disciplines, various science like computer science with molecular modeling, with the molecular visualization, from the core of chemistry till the surface of biophysics and molecular biology, along with the information of genomics and genetics. So computational biology actually opens new areas to make new discoveries. And the next slide. The research, what we do is we used to follow the molecular docking and virtual screening approach. Actually, molecular docking is an approach in which we fit molecule into 3D space, and molecular dynamics is the extension of this whole mechanism, and where we check the runtime dynamics of the simulation of the proteins. We also work on the infectious agents along with the therapeutic intervention with the algorithm development and some analytical tools. And we have certain national and international collaborative research as well. So what the protocol we follow is that we do the pocket-based virtual screening, and after that we go for a laboratory preparation along with the molecular dynamics of compounds. Either they can be a drug, they can play as a drug, and depending on the need, we go to the next step of checking them experimentally as well. So these are some comparative docking examples of our dealing with using the approach of hybrid molecular docking and virtual screening. And these are some publications which are in international journals. So where we certainly fix the problem by using that which molecule can be a potential drug. And Along with this, we go with the time-dependent dynamics of the studies of relevant human proteins, their characterizations of the therapeutic targets with unbound and bound systems to have new knowledge about those particular prodrugs. This is the algorithm development area where this is the AFD, which is an indigenously developed bioinformatic method in which we utilize the two-body interactions and in a form of a gradient formation. This was indigenously developed, while this is another method in bioinformatics which we developed with collaboration, along with the collaboration of University of Bristol with Adrian Mulholland. This method name is TUDE. So this is the AFD. This is the traditional method where we see the two particle correlation, and this is how in our AFD method we use to see and observe the interaction with and connect their functionality with having a knowledge of gradient formation of those particle interaction of those particles. So we certainly have collaborative research in which we used to collaborate with the synthetic chemist, and then we go on with the computational and the mechanism study of that particular synthetic moiety and checking them, their antibacterial potential and their drug potential. So what happens actually is we identify the disease in the drug designing. So first we identify the disease in the traditional method, and then the drug designing or vaccine designing is not a simple a two-step process. It's a process which requires different expertise at the different steps, and that's why it takes long. So we isolate that particular protein and this takes some years in the traditional method. Then finding a drug effective against that disease protein is another step which takes 2 to 5 years. And then preclinical testing and the whole scale-up processes along with the formulation till it goes to the human clinical trials, and afterwards it goes to the FDA approval. So the average design cycle is more than 15 years. While the recent trends, if you see the introduction of genomics, proteomics, bioinformatics, computational biology have actually reduced this span to a larger extent with certain new techniques like high throughput screening, virtual screening, and all this leads to the combinatorial chemistry, which is actually the chemistry of those library of those compounds. So, and this leads to the molecular modeling and on. Same is happening with the vaccine as well. So vaccine discovery is, you have to identify the disease and then you have to isolate the peptide, and this takes 2 to 5 years. And this isolation further goes with a step of the preclinical testing, and then again it goes to the formulation. And so this is the traditional method, while the introduction of new techniques in this vaccine design is reverse vaccinology and antigenicity filtration and construct design and filtrations. Various filters have been introduced to make a construct possible. And this construct, when it comes with the this filtration, whole filtration procedure, it identifies very quickly and efficiently. So, and then we can go to the synthesis to the directly and screening into the animal models. So the traditional facts are like taking a drug to 12 years or a vaccine to the 10 to 15 years in a normal cycle costs a lot and it takes a lot of time. So while in both in drug designing and in vaccine designing, the new techniques like reverse vaccinology helps in minimizing the cost in terms of human and financial resources, and people are adopting it to make a contribution to a successful discovery of various vaccines. So all of these new techniques we have applied in that work computationally, and we came with the research article which you are discussing about. And this study actually presented a comprehensive vaccinomics-based approach to complete data. We gathered that data from the NCBI and from those non-structural proteins, which are spike proteins. And epitopes were discovered, which are regions of high potential interest and having high likelihood of being recognized by the human immune system. So how we do these constructs were taken into some AI-based methods, which predicts us the titer of the antibodies, and this construct has tested. So this was a long procedure in which I'm not going into the technical details, but if you see that multi-epitope peptide-based vaccine was processed into molecular docking, and this molecular docking, when interactions pattern were recognized, we went to the series of molecular dynamic simulation and binding energy calculations. So the main point was these TLR3 and TLR4 receptor, which is important, and these constructs were stable throughout the docking and throughout the simulation, which shows that when they can be a candidate for producing immunity. So same are the results here. It is anchored to a part of this TLR4 receptor. Normally, under the vaccine constructs, we have certain limitations, and the deviations of the system is very high under the nanosecond dynamics. But if you see in the MD trajectories which we have are quite stable in these graphs, which shows that the construct which we made is actually remained stable throughout the simulation time to the TLR3 and TLR4 receptor. So these are the representation in different colors of the attachment of that receptor to the construct at different nanosecond. If you see at 5th nanosecond, at 62nd nanosecond, and 100 nanosecond, but it remains unanchored in overall the period of this simulation. This is showing the construct contact of construct at 0 nanosecond and 100 nanosecond as well. So these are different binding modes, and all the binding modes were combined to see that in all over the simulation, and after all the procedures, this construct remained unanchored and it was stable throughout the simulation. So the second part of this work was a probable prevention strategy, in which the theory of retaliation I have discussed. So because we are living under the severe threat of antimicrobial resistance as well. And together, Dr. Shinwari also discussed in his talk, and it was a very deep down analysis, a nice analysis, that animals associated viruses or bacteria are actually retaliating. So either it's whatever is the case, either it is a horizontal gene transfer, it's a transmission, but viruses are actually getting strong in infecting the host. And under this infecting the host, it's dominating the recent past. So the theory of retaliation is like the continuous attack in form of outbreaks by them have raised many challenges to us. And the dramatic impact of infectious disease affecting the modern population like us is quite evident. And in recent studies, if you see, there are multiple instances which shows that the mechanism of avian related microbes are actually advancing. So while concluding my work, because of we don't have much time, so we use the available approaches to prioritize the potential vaccine candidates in our work, bearing in mind that it will pass the general trend of those immunological applications and non-allergenicity and being antigenic and being T and B cell exposed. And these promising computational findings might deliver preliminary epitope set for a vaccine against the COVID-19, which is going under the synthesis. Meanwhile, this Homo sapiens has to avoid becoming a reason for the extinction of various species. They must have a strategy keeping in view the advancements in defense mechanism of avian and avian-related microbes avenging humans. This theory of retaliation will play its role along with maintenance of, and of course, there is a dire need of a balanced ecosystem, and which needs to be reinvestigated by the policymakers, that we have to have a balanced ecosystem. So this is my lab, and we are involved in various aspects of our computational and experimental findings. These are the awareness campaign which we have performed on antibiotic resistance within the university. These are my collaborators, Kara from Bristol and Klaus from University of Innsbruck, Rame from Harvard, Alexander Mackerel from Maryland, and Kate from Thailand. I really need to thank all of those my national collaborators and international collaborators. And last but not least, I would like to thank Professor Zabta Khan Chilwari, Professor Muhammad Ali, and Professor Amir Karam, and the PS organizers and my lab members. Thank you so much for your kind attention.